So let's start with a very high yield and frequently tested topic related to USMLE Step 1 exam. And this is a bit complex topic for the students, but I'm going to make it easy and simple, and I'm going to avoid any unnecessary or redundant detail and providing you with the solid and valuable information. So first of all, what is the purpose or function of the citric acid or Kreb cycle? So Kreb cycle connects the carbohydrate, protein, and fat metabolism with the energy production process, which is the electron transport chain. So it basically acts as a bridge between the metabolism of all these food ingredients, such as carbohydrate, protein, and fat, and then it connects the food ingredients with the process of energy generation, which is the electron transport chain. And you can see that all these, the carbohydrate, protein, fat metabolism, they all result in the production of acetyl-CoA. So this acetyl-CoA is the main reactant or main substrate uh, to carry out citric acid cycle. And this all these metabolism results in the production of acetyl-CoA. You can see that carbohydrate metabolism, it yields a molecule of acetyl-CoA. Protein, it yields acetyl-CoA. Again, fat, they also yield acetyl-CoA. And then this acetyl-CoA enters citric acid or Kreb cycle. And then there are multiple uh, intermediate, and they go through the cycle, and it results in the formation of ATPs. You can see that these are the electrons, and these electrons are generated because there are multiple substrate in the Krebs cycle. And while these intermediates are formed, it results in the production of these intermediate, or this citric acid cycle results in the formation of these electrons. And these electrons, they are being sent to electron transport chain, etc, electron transport chain. And in the electron transport chain, we use this electrical energy to convert it into chemical energy, right? And in the electron transport chain, we uh, use these electrons to convert the electrical energy into chemical energy, and the chemical energy is ATP. And then this energy is used for the various cellular function. So you can see that like TCA cycle or Kreb cycle starts from here, acetyl-CoA. And you can see that pyruvate is basically the end product of glycolysis, and this pyruvate is converted into acetyl-CoA by the action of pyruvate dehydrogenase. Pyruvate dehydrogenase is again a very high yield uh, enzyme, and it is frequently tested because if there is deficiency of pyruvate dehydrogenase or the coenzymes required for the function of pyruvate dehydrogenase complex, so it can result in the development of lactic acidosis. And if the enzyme is deficient, the patient can develop lactic acidosis. So it is important to remember. Anyways, pyruvate is converted into acetyl-CoA, and then this acetyl-CoA combines with oxaloacetate, and this results in the formation of citrate, and the enzyme is citrate synthase. Acetyl-CoA combines with oxaloacetate, and it results in the formation of citrate. And then this citrate is isomerized into isocitrate, and then this enzyme is like I'm going to encircle all the enzymes which are very high yield. So isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and yeah. So these enzymes are irreversible, and you need to remember them. So citrate is converted into isocitrate, and then this isocitrate is converted into alpha ketoglutarate by the function of isocitrate dehydrogenase, and then this alpha ketoglutarate dehydrogenase is converted into succinyl coenzyme A by the enzyme alpha ketoglutarate dehydrogenase, and then this succinyl CoA is converted into succinate, and this succinate is converted into fumarate, and then this fumarate is converted into malate, and this malate is converted into an oxaloacetate, and there is an enzyme which is known as malate dehydrogenase. So how to remember the sequence of all these intermediates? So I can give you a mnemonic, which is officer, O for officer, oxaloacetate, can citrate, officer, can I, isocitrate is for I, and then alpha ketoglutarate, keep, right? Alpha ketoglutarate, keep. So officer, can I keep selling for succinyl coenzyme A, sugar for succinate, again for fumarate, and malate, M for money. So you can remember it by officer, can I keep selling sugar for money, right? So this is how you can remember the sequence of all these intermediates. And there is another thing to remember, which is like it is frequently tested in the board exam, like which are the reactions in which NADH is produced. Which are the reactions in which NADH is produced? So you can see that uh, NADH is produced in the while isocitrate is converted into alpha ketoglutarate, and then again when alpha ketoglutarate is converted into succinyl CoA, again NADH is produced, and then there is a production of FADH2 molecule, and this FADH2 molecule is produced during the conversion of succinate into fumarate, and then there is another a molecule of NADH is produced while malate is converted into oxaloacetate. So you can see that 1, 2, and 3 molecules of NADH are produced, and 1 molecule of FADH2 is produced, and 1 molecule of GTP is produced. So you need to remember that uh, in which reactions NADH is produced, and in which reaction GTP is produced, and in which reaction FADH2 is produced. So sometime the board examiners, I know that this is somewhat uh, very nitpicky, but board exams somehow they love to test you on this concept. So you need to remember that in which reaction NADH is produced, in which reaction GTP is produced, in which reaction FADH2 is produced, and how many NADH molecules are produced, and how many GTP molecule, only 1 GTP molecule is produced, and only 1 FADH2 is produced, and 3 molecules of NADH is produced. So this is all you need to know about tricarboxylic acid or Kreb cycle or citric acid cycle.